It's Thursday, 20th of July 2023, and welcome to the Water Action Platform. This month, our theme is Pharmaceuticals in the Environment. Now, Haleon has long been a sponsor of the Water Action Platform, along with the other wonderful sponsors shown here, so it feels appropriate to engage with the topics that they grapple with in this month's broadcast. Haleon is a world-leading consumer health company with a clear purpose to deliver better everyday health with humanity. Making better everyday health more sustainable is fundamental to their business. Many of Haleon's topics are universal: carbon emissions, sustainable packaging, sustainably sourced ingredients, minimal waste. But in water stewardship terms, Haleon also care about a less common issue: pharmaceuticals in the environment. Pharmaceuticals are medicines that we use to prevent, cure, or alleviate medical conditions. Some of these medicines can end up in surface water, groundwater, and even in drinking water. This can happen when we excrete the excess after use, when we dispose of them improperly, or when they're released during manufacturing. This is a problem because pharmaceuticals can have negative effects on ecosystems. Some studies have shown that exposure to antibiotics can cause bacteria to develop resistance, making infections harder to treat. Other studies have found that hormones can disrupt the reproduction and development of fish and other aquatic animals. But despite this, we need to look at the health benefits as well as the natural environment. So today's focus areas are access to pharmaceuticals, access to clean water, and environmental protection. My first guest, Dr. Bengt Maddison, who's Policy Manager at i-phyc, the Swedish Pharmaceutical Industry Association, is here to explain.
Both are fundamental to secure long-term sustainable health. I'll show some different ways pharmaceutical substances could enter out into the environment. Very often we talk about APIs, the active pharmaceutical ingredients, coming out into the environment. And if we focus human pharmaceuticals, patient excretion, patient being treated by pharmaceuticals and they excrete substances, either mother substance or metabolites, stands for roughly 88% of the substances that we do find out in the environment. An incorrect disposal of unused medicines, roughly 10% on a global scale of the substances we have measured. And that leaves roughly 2% coming from manufacturing operations. However, it's very important to remember that those 2% could be discharged at pretty high concentrations from the manufacturing operations. So it's extremely important that we secure that we do not have unacceptable releases or discharges from manufacturing operations. The simple reason for us to not want to see the APIs out in the environment is, of course, that they are biologically active. So they are intended for a medical impact on us humans or on animals, and they will therefore potentially also have biological activity for water living organisms. It's also important to remember that many of these substances are stable. They're stable molecules. The simple reason for that is that, of course, we want them they cannot start to degrade in the body before they've done their medical impact. There has been several reports on the occurrence of active pharmaceutical ingredients out in the environment or in nature. And these reports on occurrence of pharmaceutical substances actually exist from across the globe. In many cases, very, very low concentrations. It's far below any therapeutic effects or levels. It's important to understand that occurrence does not equal a risk. On this slide, we've plotted the maximum PEC to PNIC, predicted environmental concentration, to predicted no effect concentration, which could be seen as some kind of a safe concentration. We plotted that for a number of different pharmaceutical substances. The data presents the environmental concentrations measured or predicted on the EU country with the highest exposure. And as you'll see, it is only very, very few cases where these concentrations are high enough to pose an environmental risk. Well known in this discussion is sex hormones, including ethinyl estradiol, the substance in the contraceptive pill. And I think that's a very good example where the value of access to that product is obvious to everyone. But what could we do to eliminate or limit at least any potential environmental impact or environmental risk? Next slide has some suggestions. And remembering that the vast majority of pharmaceutical substances do not pose a risk, in those cases where we see that, we need to find a way to secure access to medicine, of course, as well as access to clean water. From our point of view, responsible manufacturing, no unacceptable discharges from operations. But another perspective, of course, is what we call prudent use, responsible procurement practices, responsible prescribing, responsible disposal, and where relevant and needed, also upgrading of public wastewater treatments to be able to eliminate potentially persistent micropollutants, which would include pharmaceuticals. I'll show you some of the industry initiatives under the umbrella of EPS or eco-pharmaco stewardship. A lot of different components, as you see. Just let me shortly highlight the Meds Disposal campaign, where we look on correct or incorrect disposal of unused medicines, as well as the AMR Industry Alliance, the PSCI, Pharmaceutical Supply Chain Initiative, which all talk about manufacturing effluent management. And I really recommend you to look into those initiatives. So, in summary, the clear objective for us is to take part and a lot of collaborate initiatives and actions, which could both be legislative and non-legislative, that secure access to medicines as well as access to clean water, both being fundamental to reach long-term sustainable health. Thanks for listening.
Thank you, Bengt. It's clear that restricting access to medicines would have significant consequences for the quality of life of millions of people living with common health conditions, and restricting access could lead to an additional burden on already stretched health systems. However, as Bengt pointed out, we do need to prevent micropollutants, including pharmaceuticals, from entering the environment. Our first technology spotlight today is on i-phyc, a wastewater treatment technology which does just that. i-phyc have developed an algae-based treatment and material recovery process that can be used with a range of wastewaters and under a range of conditions. Here to tell us more about it is Simon Gretton Watson, the CEO at i-phyc.
Today I'm going to talk to you about our technology and the potential it has for tackling the emerging contaminants problem. So first of all, what is our technology? Well, we use natural microalgae within our patented photobioreactor to treat wastewater. The technology is sustainable, scalable and chemical-free, and our process can be fitted onto most wastewater sites due to its small footprint. We currently sell into 3 of the largest English municipal water companies, and our technology removes phosphorus and ammonia to consented levels. Another big benefit of our technology is the negative carbon aspect, where for every 1 kilogram of algae produced, we consume 2 kilograms of CO2. Our technology also overcomes some of the drawbacks seen in chemical treatment, where it produces a toxic chemical sludge, which requires specialist disposal, as well as the reliance on importing expensive chemicals. As well, on the nature-based side, we overcome some of the issues seen there, where they have inconsistent removal rates and require a very large footprint. Here we illustrate using real-side data the performance of our technology when removing phosphate and ammonia. As you see on the left-hand side axis of phosphate with the solid green line, we were able to take it down to 0.26 milligrams per litre within 4 hours. And for ammonia, shown on the right-hand side axis with the solid orange line, we were able to take it down to very low traceable levels within 2.5 hours. This clearly shows the rapid speed at which we can remove these pollutants. When it comes to emerging contaminants, we have made an exciting breakthrough by demonstrating the ability of the microalgae to rapidly remove emerging contaminants. We have shown that microalgae has a high affinity for removing water pollutants, and the tables show the initial results for one of our sites. As you can see, propanol, an anti-anxiety and heart medicine, was almost entirely removed, azithromycin, an antibiotic, as well as Prozac, otherwise known as fluoxetine, was also taken to very low traceable levels. Lots of other pollutants were also removed to very low levels, as shown in the table. Clearly, more research is needed, and we are looking for industry and academic partners to collaborate with to unlock this breakthrough. Please feel free to contact me if you'd like to discuss this in more detail. Thank you.
The thing I found most impressive is its proven ability to remove medicines like diclofenac with 96% removal efficiency, and this in a demonstration plant up to a really good size, 117 cubic meters per day. Thank you, Simon. So how do we know when and where we should use technologies like this one in this category, or when another option would be better? I asked an expert for her opinion on this very topic. Dr. Roberta Hofman-Caris has been working on organic micropollutants for over 15 years. She's a senior research scientist at KWR Watercycle Research Institute in the Netherlands and an associate professor at the University of Applied Sciences in Utrecht. Here's what she had to say. The first question I have for you is, how do we know whether the quality of a particular surface water body is affected by pharmaceuticals?
Well, in general, we know that it is affected because we know that, for example, wastewater treatment plants are discharging onto the surface water. And especially in the Netherlands, where the surface water has already passed so many countries with so many wastewater treatment plants, we know that it is present. But we also analyze the water because of the Water Framework Directive to check the quality. And mostly because we use the water as a source for drinking water, then you also have to know what's present.
Okay, and if the lake or the river water is being used for drinking water supplies, then how does the water need to be treated? How does it differ compared with conventional potable water treatment?
For conventional water treatment, in general you use coagulation, flocculation, sedimentation, followed by dissolved air filtration or rapid sand filtration. That should be sufficient. But in that way, you cannot remove organic micropollutants like pharmaceuticals. In that case, we have three possibilities in general. You could either remove it from water by using a membrane. You could also remove it by adsorption. General activated carbon is used for that. And the third option is that you really destroy the compounds by applying some kind of oxidation process. And there's a range of oxidation processes and advanced oxidation processes that can be applied for them.
How do you choose which technology to use? Can you predict the effectiveness of a particular treatment technology?
Yes, at the moment we can. I can say we can. We've been working in the past decade, I think, at KWR to find a way to predict the removal of organic micropollutants by certain techniques. And we base those techniques on the molecular properties of the contaminants. So by using quantitative structure property analysis, relationships, QSPRs, we can make a statistic relation between certain parameters, like the permeability of a membrane for that compound, or its adsorption behavior, or how it can be oxidized, the oxidation rate constants, the quantum yield, if photolysis is applied. We can relate those properties to the molecular characteristics of the compounds. And this can be fed into models that predict what will happen to the organic micropollutant during a certain process. And in this way, we can already predict which kind of technology may be effective And that means that we can reduce the costs of testing.
That sounds like it would work really well where there are not too many interfering compounds in the water, so good for pure water. What if there's competition from something like natural organic matter, or how does that work with wastewater, which must be much more complicated?
The matrix of wastewater is more complicated than the matrix of surface water. Although, of course, surface water consists partly of effluent from a wastewater treatment plant. So we can manage with our models quite well. But what you sometimes see is that it's not the modeling that causes the trouble. That if you want to treat wastewater, but that you first have to do a pre-treatment to improve water quality. For example, to improve the UV transmission of the water, or to remove some of the reactive organic compounds that are present in natural organic compounds. So maybe it's not that we cannot use the models, but it's more that we need a pre-treatment to make the process more effective.
And is that true for all wastewaters in general, or more for specific types of wastewaters, like hospital waste, for example, that you would expect to carry a higher load?
Well, partly. The wastewater from hospitals carries a higher load, but in general, it contributes for less than 10% to the total pharmaceutical load of surface water, because most of it comes from the communities, from the houses where people live, because they are using the medicines at home. And so more than 90% comes from the municipalities, from the towns and villages where people live. So that's not the biggest problem. On the other hand, hospital wastewater has a different composition. And we are also working on local decentralized treatment of hospital wastewater, for example, to treat that water before it enters the surface water. One of the reasons also is that you have different problems with hospital wastewater. For example, in antibiotic-resistant genes. And in hospitals, antibiotics are used as a last resort. And we certainly don't want resistance to occur for those antibiotics, because that would really be a disaster.
Thank you so much, Roberta. Roberta's group has published some landmark work on monitoring and modelling the fate of micropollutants, including pharmaceuticals, in the environment. And links to some of her work are going into the chat box now. Our next guest is Faja Musak, founder and CEO of Oxyle, and she's going to take us through our second technology spotlight today about their solution, which is a catalytic oxidation technology.
At Oxyle, our mission is to protect the health of our precious bodies of water against micropollutants using Oxyle's sustainable and innovative solutions. We specialize in removing micropollutants from water, which includes pharmaceuticals, hormones, cosmetics, and even industrial chemicals like PFAS or the forever chemicals, which, due to their widespread use and lack of currently available effective treatments, have now been found all over the world, not just in the surface waters, but also in the ground and drinking water of developed countries. This is presenting companies like Oxyle with a great opportunity to enter market with a superior technology. So what makes Oxyle's technology so much better? We have developed our innovative technology platform, which consists of our modular plug-and-play reactors that are activated by our freely available and scalable energy sources like mechanical vibrations, bubbling, aeration in the water is an energy source for our catalyst, which is composed of our patented material, which then takes this mechanical energy and converts this mechanical energy into chemical energy for redox reactions to take place, where these highly persistent micropollutants that are then broken down into harmless byproducts, basically are completely mineralized during our treatments. And we don't just say to our customers that we do our innovative treatment, we also have developed a real-time monitoring platform for our customers where we can show them in real time how well these persistent pharmaceuticals and pesticides are being degraded in real time. And we've also protected a method of treatment by filing 2 patents. So what we propose to our customers in our treatment includes installing our treatment units as close as possible to the source of problem, so basically decentralizing the problem for them, and then basically ensuring a treatment oxidizes and reduces these pollutants for them, allowing them to safely discharge the water or reuse it on customer site. Here, for example, you can see the results of our recent paid pilot study where we not only removed every single different micropollutant that was present in the customer's wastewater by over 80%, but we were also able to show very low OPEX numbers for our customers and no formation of any problematic degradation byproducts. Similarly, in this pilot, we showed not just the removal of all the different pesticides found in a customer's water, but even the removal of forever PFAS compounds to below detection limit. Hence, by switching to our technology, our customers can enjoy high micropollutant removal rate in a sustainable, cost-effective, and eco-friendly manner while reaching their discharge regulations. So far, we have completed over 10 customer projects, raised over $7 million in funding, grown to a team of 20 people, and will be launching our first products in the market by end of the year. And hopefully, with your support, we can continue to innovate bold solutions for tomorrow. Thank you very much.
Thank you, Faja. Oxyle's technology is very different from i-phyc's, but it also shows 96% removal efficiency for diclofenac and medicines like it. And I love that. The more tools we have in our toolbox to achieve the same purpose, the better. We're now going to turn our attention to microplastics. 2 or 3 decades ago, the word microplastic was hardly heard of, let alone in newspapers and mainstream media. But now it's a mainstream word, and the water sector has to figure out how to manage microplastics in the water cycle. Some of them come from personal care products like facial scrubs and exfoliants and toothpastes, and microplastics have been found in sewage samples and water bodies all over the world. Wastewater treatment removes up to 98% of microplastics from wastewater, and from there they report to the solids, the sludge. Conventional sludge digestion does not destroy microplastics. For that, we need thermal pretreatment, and that's 120 degrees C for 30 minutes. However, artificial intelligence has designed an engineered enzyme that breaks down PET, the plastic used to make things like water bottles, at the temperature of 50 degrees C, and this is both unique at the moment and perfect for thermophilic digesters. 2 weeks might seem slow, but it's 3 times faster than any other PET degrading enzyme known at the moment at that temperature. And here's how they did it. A bacterium called Ideonella sakaiensis eats polyethyl terephthalate, or PET, in the wild over weeks or months, and it breaks down the plastic using a wild-type petase, an enzyme that uses PET as a carbon source. The new enzyme, named fast petase, was designed using a freely available AI tool called Mute Compute to engineer the petase. The researchers started by taking 3 existing petases, including the wild type, and feeding each protein sequence into Mute Compute. Mute Compute looks at the positions of the amino acids within the protein, and it predicts which ones are not in fact optimized for their local environment. So Mute Compute suggests changing the positions of amino acids within the protein to make it more stable at lower temperatures particularly, and more active. The researchers basically performed a mutagenesis screen, but using AI instead of actually in a real-world lab. They computationally predicted, using Mute Compute, at every amino acid position whether a different amino acid would make the protein more active and more stable at lower temperatures. The protein mutants that were produced by Mute Compute were scored, and the ones using the best scores, or the ones generating the best scores, were then synthesized in the lab, and their enzymatic activities were determined. One variant had 38-fold higher activity at 50 degrees relative to the original enzyme, and this redesigned enzyme was named fast petase. Fast PETase still takes a long time to penetrate thick plastic, so something like a bottleneck, for example, would still take weeks and weeks. However, fast PETase in thermophilic digestion temperatures would be presented with just the right size of PETase particles to destroy them within 10 to 40 days, which is a fairly typical retention time that digesters usually have. And finally, we're going to shift from well-known, well-understood pharmaceuticals to using wastewater monitoring to discover some brand new ones. A multinational research team monitored raw sewage for 3 years. The number of countries and sites that they sampled increased from 8 countries, 12 sites in the first year, to 10 countries, 25 sites in the second year, and finally 16 countries or 47 sites in the third year. A total of 52 psychoactive compounds were analysed in total, between 26 and 34 analysed each year. Now, of course, there were spatial trends. There were different classes of psychoactive substances detected with greater and lesser frequencies and at higher and lower concentrations from place to place. The people in the 16 countries weren't all using the same psychoactive substances to the same extent. And there were also trends over time. So, for example, there was a clear impact of the COVID pandemic. Lower concentrations of psychoactive substances in sewage unsurprisingly coincided with lockdowns when people weren't able to get together and socialize. Most interesting, though, was that some metabolites detected in the sewage revealed 18 entirely new psychoactive substances, some of which had not been invented by the formal pharmaceutical or chemical industries. These substances aren't illegal, but only because they were unknown to science and not regulated yet. And with that, we come to the end of this month's broadcast. I once again thank all of our contributors and sponsors, especially Haleon. And next month, we will be hosted by Alicia Seil. She's going to focus on young people's achievements in the water sector, especially in emerging economies and the global south. And I really hope that you'll join us then. Until then, keep asking questions, keep sharing, and keep safe.